We introduce you a 21-year-old woman who lives alone and have a rich diet in starch, meat, ham, sausages, cow, and goat dairy products like fresh cheese, vanilla cheese, lactose-free milk, yogurt, and goat cheese. This woman also suffers from seasonal allergies that manifest with a constant flow of nasal mucus, hyaline, itching at the throat, and eyes with constant tearing. One day she started to feel sick. Her discomfort escalated until she got really sick and the physician asked her to take some tests, in which she gave a positive result to Brucella and some other pathogens, but we are focusing on Brucella. Brucellosis is a bacterial disease caused by various Brucella species, which mainly infect cattle, swine, goats, sheep, and dogs. Most cases of human brucellosis are caused by ingesting unpasteurized milk or cheese from infected goats or sheep. The genus Brucella comprises several bacteria species that can cause brucellosis. However, the agents that most frequently cause human brucellosis are B. melentensis and B. abortus. Brucellosis typically causes flu-like symptoms, including fever, weakness, malaise, and weight loss. The incubation period of the disease can be highly variable, ranging from 1 week to 2 months, but usually 2 to 4 weeks. Brucella is an aerobic gram-negative Coccobacilli bacteria. Brucella can infect humans by affecting the immune system in several ways. Brucella has several strategies to establish and maintain a chronic infection, including inhibition of apoptosis of infected mononuclear cells, prevention of dendritic cell maturation, reduced antigen presentation, and reduced activation of naive T cells. We can assure that the woman got Brucella from the goat dairy product she likes to eat, so the first site of colonization must have been the TGA. Brucella is capable of surviving the acidic environment of the stomach and can pass through the stomach to reach the lower parts of the digestive system, such as the small intestine. One important characteristic of Brucella is that it is a stealth invader. Brucella enters the host cells without apparent activation of the innate immune response through toll-like receptor ligand interaction. Phosphatidyl dinositol triphosphate (PI3K) promotes binding of toll-EL1 receptor tear-containing proteins on the plasma membrane and subsequent TLR signaling. Brucella prevents this by expressing at least 2 tear-containing proteins to ultimately restrict the pro-inflammatory response and dendritic cell maturation. To delay being recognized by the innate immune response and initiating an immune response, Brucella reduce, modify, or cloak their pathogen-associated molecular patterns, PAMPs. Also, strains with lipopolysaccharide (LPS) are able to restrain host cells' apoptosis by the interaction of the O change with TNF-alpha, tumor necrosis factor alpha, so dead cells do not release specific factors, do not activate the immune system, and Brucella are able to avoid host immune surveillance. Brucella displays strong tissue tropism, which is the ability to infect specific types of host cells within an organism. Brucella deploys the T4SS type 4 secretion system, a complex molecular machine, to facilitate its entry into the host cell. This system allows the bacteria to directly inject effector proteins into the host cell cytoplasm. These effector proteins control the intracellular stealthy lifestyle of the pathogen. Once Brucella enters into the host cells, it starts to reproduce and break the cells when there is no more space to proliferate. When this happens, the first cells to arrive are macrophages. Macrophages engulf bacteria to destroy it, but Brucella bacilli can survive inside macrophage. Inside mononuclear phagocytic cells, Brucella reside in a special vacuole, Brucella containing vacuole BCB, modify intracellular trafficking and transform the vacuole into a replicated compartment or Brucellosome. Brucella evades intracellular destruction by restricting fusion of the BCB with the lysosomal compartment. The same as in normal cells, when Brucella breaks the macrophage, promotes the release of cytokines such as EL1, EL2, and interferon gamma produced by Th1, T helper 1 lymphocytes, that triggers the humoral response. Humoral response only kills extracellular Brucella, and Th1 kills intracellular Brucella. The parts of the host cells that are left trigger the action of T cytotoxic lymphocytes. Cytokines are key players in the protection against brucellosis, mediating both innate and adaptive immune responses. IL-12 produced by B cells and macrophage leads to a Th1 response and induction of interferon gamma, which activates macrophage. The main alternative therapy against brucellosis is doxycycline plus rifampicin for 45 days. Also, there is some vaccine to prevent infection in animals, but what if there is no vaccine or antibiotics for the treatment?
The search for new approaches in therapies against pathogenic bacteria has led to the creation of treatments with molecules other than the antibiotics that are known. In this way, a therapy using nucleic acids sounds very interesting, especially if it helps eliminate an infection, such as brucellosis. Peptide nucleic acids, PNA, are analogous molecules of single strand. Synthetic nucleic acids that contain as a skeleton a vertebral column based on the formation of peptide bonds instead of the phosphodiester sugar phosphate bond. PNAs have the ability to bind in a complementary way with the strands of DNA and or RNA because in the structure are the purine bases, adenine and guanine, and pyrimidinic bases, cytosine and thymine, such as in nucleic acids. The DNA-PNA bond is even stronger than that of DNA-DNA, since the PNA does not contain ionized phosphates, so there is no electrostatic repulsion of negative charges. And how would PNAs help eliminate brucellosis? Being molecules analogous to DNA, their molecular target will be par excellence the bacterial DNA of Brucella, specifically genetic sequence involved in different types of macromolecular synthesis: DNA, RNA, lipids, nuclear envelope, proteins. These genes have to be essential for both the functioning and growth of Brucella, since what is sought is to inhibit its development. The genes chosen then are those that code for proteins such as DNA polymerase or bacterial RNA polymerase. The complementary bond between the bases of Brucellosis, while DNA and the PNAs will interrupt the gene so that the synthesis of the protein will not take place.
In this video we make a proposal that involves an alternative treatment for Brucella SP with other microorganisms, imagining that there were no vaccines or antibiotics. We will use an engineered probiotic or microbiota bacteria. Brucella SP has the ability to evade the immune system by hiding inside the host cells, adhering to the endoplasmic reticle and generating the Brucella-containing vacuoles using the secretion system type 4, in this case named VIRB. Brucella SP is a problematic pathogen because it can remain inside the macrophages and other immune system cells, using them as a transport through the lymphatic system and bloodstream until these cells burst, releasing bacteria and cytokines. Then the immune response is triggered using T and B cells, but they can only attack extracellular bacteria or those who were released from burst cells, leaving intracellular bacteria untouched. These survivors enter a latency period, waiting for the immune response to weaken and cause a new infection. What we propose is to modify a microbiota's cell with genetic engineering to block the secretion system type 4 VIRB for Brucella, through the modification of a plasmid belonging to a natural microbiota bacteria from the host or any probiotic cell, such as Lactobacillus or E. coli, capable to conjugate genetic material between the cell and the pathogen. The idea is to modify the gene sequence which express any of the next proteins, VIRB4, VIRB11 or VIRB6. The lack of these proteins would stop the production of Brucella-containing vacuoles. Therefore, Brucella won't be capable of remaining inside the cells. In addition to this, we could apply the same treatment, but this time to promote the secretion of interleukins and gamma interferons to generate an immune response with Th1 cells and B cells. The problem with this is that we could generate an overreaction of the immune response. For our clinical case, the treatment could be mixing our viable bacteria in an ampoules, granules, or soluble tablets with the milk and yogurt that our patient consumes a lot before their ingestion.